Uh, Thank you very much, participants and the respected dignitaries, for being patient and cooperative with us. Our next speaker, Dr. Sachin Kumar, is in line with us. Let me give you a brief introduction of biography of Dr. Sachin Kumar. Dr. Sachin Kumar is Deputy Director in Biochemical Energy Division at the Sardar Sawan Singh National Institute of Bioenergy, Kapurthala, India. He was a visiting professor in Department of Chemical and Biological Engineering at South Dakota School of Mines and Technology, Rapid City, USA. For a period of 1 year, he obtained his PhD in Chemical Engineering from Indian Institute of Technology, Roorkee, India, and has more than 17 years of research experience in biochemical conversion of biomass to biofuels, including lignocellulosic, ethanol, biogas, biohydrogen, etc. He has completed 8 research projects and 1 consultancy project and actively engaged in ongoing research projects. Dr. Sachin has published more than 60 papers in peer-reviewed journals, book chapters, and papers in conference proceedings, and 7 edited books. He has 1 granted US patent and 1 filed Indian patent. He has delivered more than 40 invited plenary lectures and presented more than 75 papers in national and international conferences. He is the recipient of 2016 ASM IU SSTF Indo-US Research Professorship and selected as Bioenergy Award for Cutting Edge Research Fellow 2016 by DBT and IUSSTF. So, we are very esteemed to have such an eminent speaker in the field of bioenergy with us. Very warm welcome to you, to Dr. Sachin Kumar, for being associated with the FDP of University of Lucknow and having privilege with you for giving your esteemed time to Lucknow University to give an invited talk on bioenergy, sir. I am making you the host now, so you can give your presentation to the attendees and the dignitaries. The stage is set for your presentation, and thank you very much and warm welcome to you on the FDP, sir.
Yeah, thank
I
you.
made you the
Thank
host,
you.
sir.
Thank you for the nice introduction. And I'm very much thankful to the organizers, mainly Dr. Neelam Mishra, who invited me to deliver this talk. Actually, she is also alumni of IIT Roorkee. So we had this collaboration, initial collaboration, and hope for the future collaborations as well. So thank you organizers once again. So let me share my presentation. Okay, is it visible?
Yes, sir. Your screen is visible, sir. You can go on. Thank you very much, sir, for your presence.
Okay, thank you so much. So let me start. So I will give a talk on bioenergy conversion technologies. So what are the prospects and challenges? So as we all are aware, the current scenario worldwide, we are mainly dependent on the fossil fuels or the conventional energy. Now the time to shift the paradigm to non-conventional energy. So bioenergy is one of them. Okay, so if we talk about the biofuels, just a minute, let me short this window. Okay. All right. So if we talk about biofuels, so the fuels whose energy is derived from the biological carbon fixation. So biological carbon fixation is just the growth of plants. So through that we get the biomass. At that using that biomass, the fuel which we generate, that we can call biofuels. So in line with the conventional fuels, we can categorize biofuels as well, like liquid biofuels. So if we talk about liquid biofuels, they are like bioethanol, bioethanol, biodiesel. So these liquid biofuels we can blend with the uh, transportation fuel. Like we can blend ethanol and butanol in petrol or gasoline, or biodiesel we can blend in the diesel and can be used as a transportation fuel. Next, we can categorize gaseous biofuels. So in gaseous biofuels, biogas, biohydrogen, producer gas. So there are different applications, including the heating in the households or in kitchen, and we can also produce electricity using these gaseous biofuels. So biogas we can further upgrade to bio CNG and can be used in the transportation, and we can also feed into the grid to generate the electricity. Similarly, biohydrogen we can use in transportation and other applications. Producer gas we can directly use for the heating as well as for electricity production through which we generally generate through the gasification. The third category we can have solid biofuels. So as we all are aware, the woody biomass which is already dense, so we can use them directly for the heating applications. But there are some loose biomass. If we talk about like agricultural waste, so that agricultural waste, if we use them directly, then we cannot have the we cannot use or utilize it efficiently. So if we the reason behind it, the low density. So the disadvantages of low density is we need to put more energy to transport from one place to another place, and even we don't get the sufficient efficiency of even in the combustion. So the best application may be we can densify them. So through densification, we can produce briquettes and pallets. So they we can use them directly. Excuse me. Sorry. Okay, so we can use these briquettes and pallets in the boilers, biomass cooking stoves, and even in the gasifiers. We can use. Presently, government has mandated 10% blending of these pellets or agricultural waste basically to feed along with the coal in the NTPC power plants. So they are demanding for mainly pellets from produced through the these agricultural waste. Okay, if we see the worldwide energy shares, so this is the picture from 2016. So we see the energy utilization, mainly energy coming from the fossil fuels, which is around 80%. And then rest 20% accounts partial nuclear energy, traditional biomass like 7.8%, and then mainly renewable, modern renewables, they that accounts around 10%. Okay, so it includes like wind, solar, biomass, geothermal, ocean power, and biofuels for transportation, hydropower, biomass, solar, geothermal, heat. So that accounts for the modern renewables. Okay, based on the feedstocks, we can categorize the different generations of biofuels. So, first generations comes from the biofuels which are produced through sugary or starchy materials. Right, sugary means the sugarcane juice, beetroot, or starchy materials like grains, like corn and wheat, rye. So they come under the first generation biofuel. Even the biodiesel, the vegetable oil which we use for the biodiesel production, that comes under the first generation biofuels. But now, because they all are mostly food crops, so there are different issues. So in 2008, there was a hike in the food prices globally, which created a big controversy. And in a 13th Regional Conference of Food and Agriculture Organization in Brasilia. There was a special rapporteur from UN, Jean Ziegler. He called it crime against humanity. And since then, or during that time, the researchers and I mean, the people in the market started thinking about the alternatives or alternative feedstocks to generate these biofuels. So then this term came second generation biofuel. So that accounts mainly feedstocks from waste part of the crops. And that may be like rice straw, sugarcane bagasse, wheat straw, corn stover, corn cob. Wood waste also comes under the second generation. So mainly the lignocellulosic part, whether it is from the crop or any other plant, that comes under the second generation biofuels. In the similar line, if we see our earth that is covered, I mean, two-third of earth is covered by the water. So we have only one third of land, which we need to use for crop production and other plantations. So we cannot dependent on the second generation only. So that the two-third part which is covered by the water can also be utilized to generate feedstocks for biofuel production. So that comes under the third generation category. So the first name came for the third generation or the water grown plantations or vegetations, that is algae. So algae is considered as a low cost, high energy and renewable feedstock, and it having the potential to produce more energy per acre if we compare with the land crops or conventional crops. In the similar line, there is a fourth generation biofuels. So in fourth generation biofuels, they are, I mean, we talk about the engineering of that may be the plant or maybe the microorganisms or I mean whatever we are doing some genetic modifications or engineered organism or plantations, that comes under the fourth generation category. So through this engineering, we can do, like if we talk about a biomass, so that contains cellulose, hemicellulose, lignin. So lignin is that entity which creates the hindrance to utilize cellulosic and hemicellulosic part. So through engineering, we can decrease the lignin, or increase the cellulosic part, or we can change the crystallinity of cellulose, hemicellulose. So the hindrances which are in the utilization of that biomass can be overcome by engineering of those plants. Right. Similarly, the microorganisms, there are some organisms which can utilize cellulose, but they can't utilize hemicellulose. So we do engineering to modify them so that they can utilize both cellulose and hemicellulose. So such processes or the methodologies we use, that comes under the fourth generation biofuels. Another thing, CO2 sequestration. So the CO2 generated either through the industrialization or industries or through transportation, that CO2 we can capture it. Uh, I mean, automatically, not automatically, conventionally, which is utilized by the plants and can be converted into the biomass. That is the natural process. But the man-made or some capturing and converting into useful products can also be done. So that is also under the fourth generation category. Okay, so if we talk about biofuels, so why we need biofuels and what are the resources? So the first concern is environmental safety. If we talk about the conventional fuels, that is mainly fossil fuel, whether it is coal or the crude oil, which we are importing from the other countries like Gulf countries. Okay, So these fossil fuels, they are coming from crust of the earth, and we are getting on the surface and burning it. So this is the addition. Whether it is producing CO2 or any other toxic gases are generated through its utilization. So we are adding to the environment. So they are mainly greenhouse gases. Okay, so environmental safety is the first concern. If we replace partial of them, fossil fuels, we can reduce a lot of greenhouse gases. So the using biofuels, because they are carbon neutral, they are capturing CO2 from the environment, and they are doing their growth, and that biomass, if we utilize it, the same amount of CO2 will be released after its digestion or combustion. So they will be the net zero addition to the environment. So they are carbon neutral, reduction in greenhouse gases. Second is the energy security. So as I told you earlier, we are dependent on Gulf countries to import of this fossil fuels or crude oil. Okay, so because we have plenty of biomass or plantation of that vegetation in country that is evenly distributed throughout the country. So that by utilizing that biomass or that resources, we can be energy secure, independent from crude oil. Yeah, we are already in abundance to lignocellulosic waste, and yeah, the important part, in some places like Punjab or UP or Haryana, the residue which is left over after harvesting the crop, that is burned over there. So that is, we are losing such an important resource by burning it. And again, that damages the soil fertility, and of course environment is affected by that. So by utilizing these biomass, we can manage that waste properly. All right. So there are different conversion routes from biomass. So we can categorize like biochemical processes, thermochemical processes. So in biochemical process, because it's a like complex structure, so we need to go through the pre-treatment. So in the next slide, I will explain it. And then we follow enzymatic hydrolysis, followed by fermentation to convert into ethanol, butanol, hydrogen. So that is the like biological processes. Whatever residue, we can further utilize them either through the thermochemical process or some catalytic processes uh, to convert into other products. Second is the anaerobic digestion. So this is they are a consortium of microbes or bacteria, which is mainly responsible for this digestion and produces biogas. And that biogas, we can further purify it and can be used as a bio-CNG. All right, and second process we can categorize thermochemical processes. So they are like gasification, pyrolysis, hydrothermal liquefaction. So biomass we can use directly in gasification, and it is converted into the syngas or producer gas. So producer gas we can use directly for heating, and can also be used for can be used in the engines. They are modified engines available in the market which can directly produce electricity out of this syngas. There are different processes available which I mean there are some bacteria are available which can utilize the syngas to produce ethanol and other organic acids, and through catalytic synthesis we can also produce methane, Fischer-Tropsch diesel, dimethyl ether, methanol. Okay, similarly there is a pyrolysis process where the most of the biomass is converted into bio oil, and that bio oil we can treat it hydrologically and refine into hydrocarbon. Okay, and similarly there is a hydrothermal liquefaction either in the presence of water or in presence of hydrogen. we can convert into the bio oil. So these are the thermochemical processes. Okay, now we talk about biorefinery concept. Okay, if we go for a single product production, then that may not be economically feasible. So what we do, we have planned to utilize that biomass in a biorefinery manner. Biorefinery means if we talk about conventional refinery, so what we do, we refine the crude oil and produce multiple products, not only fuels like gasoline, diesel, or kerosene, or jet fuel. There are some higher carbons also there. So that is called like bags, so which is can further be refined to multiple products. So that's why it is called a refinery. So in a similar manner, we can utilize biomass to produce multiple products. They may also be the intermediates to some other products like. Okay, so some processes are given over here. So we can categorize in primary and secondary one. So anaerobic digestion or gasification, they are used to produce gas. That may be the producer gas or biogas or methane. So these gases can further be combust to produce heat or power, either through the boilers or through there are engines available where we can feed it and can produce electricity directly. Okay, there are some catalytic conversion processes or fermentation where we can produce hydrogen, diesel fuel, jet fuel, methanol, dimethyl ether, ethanol. All right. Uh, Through different route like pretreatment and fractionation, followed by enzymatic hydrolysis and fermentation, we can produce ethanol, butanol, lactic acid, levulinic acid, furfural, hydroxymethyl furfural. Uh, so multiple products we can produce. And this lignin part, that is the people are working on it. Cellulose and hemicellulose part is easy to digest, but lignin it's difficult to digest through microbially or even catalytically. Okay, but I mean, some people or researchers have developed some catalyst and a process to utilize it, whether it is a thermochemical process or enzymatic process. So it can be used or can be used to produce multiple products. So I have next slide where I will elaborate what products we can generate through lignin. Through liquefaction and pyrolysis, we can produce bio oil or bio crude, which can be further upgraded through cracking or hydrotreatment to produce surfactants, fuel additives, adhesives, resins, phenolates like that. So there are some value-added products from lignin. So there are basically three groups in lignin. One is the coniferyl alcohol, another is the synapyl alcohol, then paracumaryl alcohol. These further can be defined into products like vanillin, guancol, syringaldehyde, syringol. They having different applications and very demanding in the market, whether it is in the confectionery or pharmaceuticals. So they can be used as a like flavoring agent, chemical and pharmaceutical precursors, smoke flavor, uh, hair and fiber dyes, uh, preservatives like that. All right. So you can see in this slide there are common ethanol blends. We generally call gasohols, like E5, where we blend 5% anhydrous ethanol and 95% gasoline. So we will call it E5 blend. Similarly, if we increase the ethanol concentration, that terminology will change like E10, E15, E25. So like up to 25%, there is no need to modify the engines. We can use this E25 blend in a existing engines. But if we go beyond this blend, then we may need to modify the engines as well. There are two popular bonds. blend, sorry. So E85, where 85% anhydrous ethanol and 15% gasoline is used. So this blend is generally used in the USA, and they are a special kind of cars available in the market where we can use this type of blend. But this blend E100, this is the most economical blend where we are not adding any gasoline. It's the pure ethanol. Even 5% water is there. So 95% ethanol is, this is the 95% ethanol, which we also call azeotropic mixture. Okay, why we call it azeotropic mixture? Because when we do distillation to the broth, it it can be refined up to 95% ethanol only, and that blend is called the azeotropic mixture. But for blending in the gasoline, we need to have anhydrous ethanol. So we need to go through the secondary process to get this anhydrous ethanol. So that may be the secondary distillation, like tertiary distillation or azeotropic distillation, or some solvent extraction or membrane processes can also be followed. So I mean, there are two steps involved to get it anhydrous ethanol. But this E100 blend, there is only single step needed to get it, and we are not blending any gasoline over here. So this is the most economical blend which can be used in the cars. So the special cars like flex cars or flex vehicles are we can use this blend in that cars, and it is mostly practiced in the Brazil, right? Now, this picture showing different blends globally. Okay, this picture showing how ethanol is carbon-carbon neutral. So, for any photosynthesis, there is 6 moles of CO2 plus 6 moles of water are required to produce 1 mole of glucose. And that 1 mole of glucose is fermented. It produces 2 moles of ethanol and 2 moles of CO2. And in the engine or in the combustion chamber, this 2 moles of ethanol will produce 4 moles of CO2 and 6 moles of water. Okay, so overall, we are getting 6 moles of CO2 out of fermentation as well as combustion. And this again, 6 moles of CO2 will go to into the photosynthesis. So in a loop, if we see, there is no emission in the environment if we use biomass. All right. I mean, our country or India is rich in agriculture. So, this picture showing different crop residues generated worldwide, and this is divided into continents, like yellow showing Europe and Asia, America, Africa. So mainly residues are like barley, maize, paddy straw, soybean, sugarcane, wheat. uh, if we see this paddy straw part, so Asia is the largest producer of this paddy, and then this is wheat or maize, then sugarcane. So we have plenty of residues available. So this is in the million metric tons. This is for per annum. Now we see in the next slide how paddy straw or paddy production is distributed. So we are the second largest producer of paddy or paddy straw. So we almost generate more than 200 million metric tons of paddy straw per year. So this is the table showing residue generation state-wise. So the first, the largest producer of residues is Uttar Pradesh, then Punjab, Maharashtra, Andhra Pradesh. So total residue generation per year is around more than 500 million metric tons. But now question arises. they are different applications. Existing applications are practiced to utilize that biomass, so we cannot compromise with that biomass, I mean those applications. So the residue which is available in the surplus can only be utilized for the biofuel generation. So they are like residues around more than 140 million metric tons of residues are available per annum that can be used for biofuel production. Okay, so this is the process for lignocellulosic biomass to ethanol. Uh, you can see the structure of this lignocellulosic biomass. We go through the pretreatment, and through that pretreatment, we go for the enzymatic or acid hydrolysis. Then, if suppose we are going through the acid hydrolysis, we need to do the chromatography, or we need to recover the acid so that we can recycle it. Then only it will be economically feasible. Uh, and once we get the fermentable sugars, we do fermentation and can produce the ethanol. So if we see the technical challenges involved due to the complex structure of this lignocellulosic biomass, we go through the pre-treatment. You can see here in this picture, this blue fibrils, they are basically celluloses, and it is covered with this hemicellulose, this green part. Okay, that orange or this pink one, they are like the bonds of esters, and they make it very compact, and it is covered with the lignin. Okay, so to utilize these cellulose fibrils, we need to uncover this biomass. First, we have to remove the lignin, then hemicellulose, and to break down these esters. So to do this practice, we need to go through the pre-treatment. And if we talk about pre-treatment, it accounts around more than 30% of the process cost in the pre-treatment. Another is the separate saccharification and fermentation. So these two processes are biological one, saccharification and fermentation. And due to the difference in temperature and other conditions, we need to process separately. Another challenge is utilization of both cellulose and hemicellulose. So cellulose, that is like a polymer of hydrous sugars or mainly glucose. So there are a lot of microbes or even industrial yeast, that is Saccharomyces, that can utilize glucose, the hemicellulose, which is a polymer of multiple sugars, but mainly xylose. So that Saccharomyces can't utilize that xylose part. So this is another challenge in this fermentation. So we will take it one by one. Pre-treatment. So there are different pre-treatment methods. So we can categorize in like physical, chemical, biological, or combination of above. So in the crop residue, which is already large in the I mean large in the size. So we need to reduce the size to increase the surface area. Okay. So we can follow the milling or grinding like ball mill, Vibro energy, two roll pressure hammer like that. And there are different other methods like irradiation using electron beams, gamma rays, microwaves. There are different processes like high temperature processes like pyrolysis, steam explosion. They all come under the physical method, and they mainly increase the specific surface area and size of the pores, so that if we go for the enzymatic process, so enzymes can be accessible, or the cellulose can be accessible to the enzymes so that it can hydrolyze it. They also decrease the degree of polymerization of cellulose and its crystallinity, hydrolysis of hemicellulose, partial repolymerization of lignin. The second method we can follow, that is a chemical one, where we can use alkalis like NaOH, alkaline H2O2, or liquid ammonia. They are different acids can be used like H2SO4. There are some gases like SO2 can also be used, oxidizers, reducers, organic solvents, SO2 catalyzed steam explosion, ammonia fiber explosion, ionic liquids. So they affect they mainly delignify the biomass. They also decrease the degree of polymerization, crystallinity of cellulose. They increases its swelling or growth of the porosity. There are different fungi, mainly white rot fungi are available in the environment, or that they can degrade the biomass. They are mainly degrade the lignin part. So uh, they can be used as one of the method for pretreatment. They also reduce the degree of polymerization of cellulose and hemicellulose. Okay, so one method cannot be effective. So we can go in the combination like physical followed by any chemical or biological method, like alkali pulping associated with steam explosion, grinding followed by alkaline or acid pretreatment. So that is again degrade the hemicellulosic part, they delignify or increases the specific surface area and pore size. Okay, now come to the hydrolysis part. So hydrolysis may be like acid or enzymatic one. So in acid, most preferred acid is sulfuric acid. So we can categorize based on its concentration into two, like dilute acid hydrolysis and concentrated acid hydrolysis. So if acid concentration is less than 3%, we call dilute acid hydrolysis, and it need to be performed part is solubilized. And then in the second case or second stage, we keep the harsh conditions like high temperature, high pressure. So most of the cellulose is solubilized in the second stage. Even at the high temperature and pressure and harsh conditions, the recovery is quite low and cost is very high. So we generally do not prefer this dilute acid hydrolysis. Now second is concentrated acid hydrolysis, where we generally use more than 3% acid, sulfuric acid. In this case, most of the pentose or hexose sugars are solubilized, and sugar recovery is higher. But this process will be cost effective only when we recover the acid and recycle it for the next process. Okay, now come to the enzymatic hydrolysis. So they are there are multiple organisms that may be like bacteria or fungi available in the in an environment which can be used to, or they are able to produce the enzymes available. Excuse sorry. So they are available, which we can use enzymes after producing through those organisms, and can be used as a enzymatic conversion, or directly microbes can be utilized, which is called direct microbial conversion. Okay, so the enzymes involved, they are like cellulases, hemicellulases, and ligninases, and yeah, we can further modify them to increase the efficiency. All right, but for the enzymatic process, the pre-treatment is must, so that we can expose cellulose to the interaction with the enzymes. Okay, so these are three enzymes involved. if we talk about the hemicellulases, so hemicellulose, that is like polymer of multiple sugars, but main sugar is xylose, but there are different sugars like arabinose, glucose, galactose, mannose. So they are again linked with the side chains. So you can see in this picture that yellow part, they are mainly C5 sugars, mainly xylose, and they are side chains with maybe like galactose, arabinose, mannose, galactose, like that. So actually enzymes, they are very specific to break down any bonding. So each bonding is specific, so we need multiple enzymes to break down completely. So you can see the list of enzymes involved to so that's why we call it hemicellulases. So they may be like 1,4 endo 1,4 beta xylanase, beta d xylosidase, like arabinofurosidase. So there are multiple enzymes involved in hemicellulose digestion. Similarly, in the cellulases, uh, it 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 is again a group of enzymes involved, like endoglucanase, cellobiohydrolase, beta glucosidase. Then only we get glucose as a final product. So you can see the first endoglucanase acts on the cellulose fibrils to break down into like a degree of polymerization around more than 6, and then these shallow biohydrolysis acts to decrease further to obtain the shallow bios, and the beta glucosidase that acts on the shallow bios to break down into the glucose units. Okay, now the next step is fermentation, where the lot of bacteria and yeast are available, which utilize glucose and xylose to convert it into ethanol. But again, there are issues. That issue may be like fermentation efficiency, fermentation yield, or ethanol yield, productivity, utilization of both glucose and xylose simultaneously. And no natural organism is available which is perfectly fine to fulfill the requirement. So these are the key pathways which are involved in xylose utilization. If we see the pathway in the yeast, which follow this XRXDH pathway, or we also called oxidative reductive oxidative pathway. So here, first enzyme which is involved, uh, xylose reductase, and then further xylitol dehydrogenase, and then xylulose kinase. Then this xylulose 5-phosphate is, it enters into the glycolysis pathway only. And the second one, that is xylose isomerase pathway, where only xylose isomerase enzyme is involved, followed by xylulose kinase. So this xylulose kinase is mostly common one. And even this xylulose kinase is available in the Saccharomyces. But these excuse me. These two enzymes, xylose reductase and xylitol dehydrogenase, are not there. So we can do some engineering, or we can do insert these genes so that they can utilize the xylose. So a lot of work has been done over here to make the organism which can utilize only glucose, and which is modified to utilize xylose as well for the ethanol production. All right. So what is the current trend? Metabolic pathway engineering or genetic engineering is one, which is used to make consolidated bioprocessing. So what is consolidated bioprocessing? And the single organism can be used to produce the enzymes so that the polymers can be hydrolyzed, and the same organism is able to ferment it as well. I mean the glucose and xylose, which is hydrolyzed product and can be utilized by the same organism to produce ethanol. So this picture will give you the clear view. We see the enzyme production, and then we go for the enzymatic hydrolysis, then hexose fermentation, one separate part, then pentose fermentation. If we combine this hexose fermentation and pentose fermentation, we call it co-fermentation. If we combine this hexose fermentation with enzymatic hydrolysis, we call simultaneous saccharification and fermentation. Same with the pentose fermentation, we call simultaneous saccharification and fermentation. But if we combine enzymatic hydrolysis with co-fermentation, we call simultaneous saccharification and co-fermentation. But if we have an organism which can produce the enzyme, can hydrolyze it, hydrolyze the biomass, and can ferment both the hexose and pentose sugars, we call consolidated bioprocessing. Or the organism which is able to do so, we call consolidated bioprocessing microorganism. Okay, second biofuel we can consider biogas. Where again we can utilize multiple feedstocks like lignocellulosic biomass, proteins, lipids, any carbohydrates. So again they need to hydrolyze it. So in the anaerobic digestion, mainly a group of bacteria or one microorganism is involved. We call it consortium. So there are some hydrolytic microorganisms which produces enzymes which can hydrolyze those carbohydrates or proteins or lipids to monomers like sugars, amino acids, and glycerol. The second category of microorganisms involved in fermentation or acidogenesis, which produces alcohols or long chain organic acids or fatty acids, along with carbon monoxide, ammonia, and hydrogen. And then in the third stage, the acetogenic microorganisms are involved, which utilize these alcohols and long chain organic acids to produce acetic acid, hydrogen, and CO2. Okay, now we have CO2, hydrogen, and acetic acid, and that they are utilized by there are two kind of archaea. One is hydrogenotropic and another is acetoclastic one. So acetoclastic one utilize acetic acid to produce methane and CO2. On the other hand, hydrogenotropes utilize hydrogen and CO2 to produce methane. So the synergy between these hydrogenotropes and acetoclast decide the concentration or final concentration of methane in the biogas. If we see the rate limiting reactions, so hydrolysis is one of them, and then the methanogenesis. They both reactions are rate limiting one. If we see the theoretical quantity or composition of biomass, so carbohydrate form around 0.38 meter cube per kg of biogas with 50% methane and CO2 composition, and fat gives the maximum biogas yield with 1 meter cube per kg of volatile solid with even the maximum methane composition of 70%. Proteins give 0.53%, 0.53 meter cube per kg biogas with 60% methane content. So there are different substrates. The approximate methane yield is given over here. So in food waste, fruit, vegetable waste, manure may be from cattle, pigs, or chickens. Slaughterhouse waste, cereals, sugar beets, silage grasses, straws, municipal sludge waste, distillery waste, or any effluent. They can be used for biogas generation. Okay. Uh, now come to some microbiology. They are three. I mean, based on the growth temperature, we can categorize microbes in three categories. One is psychrophilic, where the organism can grow below 20 degrees centigrade or below 15 degrees centigrade, and below 40 or 45 degrees centigrade we call mesophiles. I mean, between 20 to 40 we call mesophiles, and more than 40 or 45 we call thermophiles. Okay, so sorry. Okay, now you can see in this picture the growth rate again varies with the temperature or these microorganisms. So in psychrophilic, the growth rate is very least growth rate is there, and in mesophiles it's a moderate one, and then in thermophiles the highest growth rate is there. So we can utilize these benefits of thermophiles in the biofuel production. So the main benefits are like fast growth rates, high metabolic rate of digestion. Due to this high rate, there is a short HRT. At high temperature, there is a low risk of contamination, and maybe there is no pathogens available at high temperature there. Increased conversion rates, which lead to high biogas yields. Due to the short HRT, the smaller reactor volume would be required. Most of the thermophiles having the capability to produce cellulases, hemicellulases, so they are again helpful in degradation of biomass or lignocellulosic biomass. If we compare the biogas production between mesophilic and thermophilic, so you can see here around the maximum biogas we can get out of mesophiles, that is 100 meter cube per day. But in case of thermophiles, we can have around more than 120 meter cube per day biogas production. Okay, along with the advantages, there are several disadvantages as well. So they are, I mean, additional cost would be required to maintain high temperature, but that can be maintained mm, you can do insulation. And one more thing, most of the microbial reactions, they are exothermic, so you can control temperature in the reactor. Even very little energy would be required to maintain the temperature. Yeah, they are sensitive to temperature and pH. A little fluctuation in temperature or pH would affect the its growth or the production, whether it is biogas or bioethanol. Liquid quality getting worse due to the high mass conversion. Yeah, that is true. Okay, so this is one example for the paddy straw. So in paddy straw, it's like a rigid structure. It needs to be pre-treated. So again, there are different pre-treatments which we discussed in the last slides. Okay, if we talk about the organic matter, which is around 70 to 82%, then theoretical biogas we can have from out of this paddy straw. This is 700 liters per kg of volatile solid, and experimentally we can have 50 to 280 liters per kg volatile solid, which is around a maximum 40% of the theoretical yield. But we can increase this biogas yield by using like pre-treatments. We can increase up to 525 with HRT like 30 to 65 days. But the drawback is it is expensive and spacious. Another terminology which we need to maintain, that is the C/N ratio, which we can overcome by co-digestion or some chemicals which are rich in nitrogen can be used. Another issue, silica. So silica can be overcome through pretreatment only. But you know, pretreatment is the expensive one. So how we can avoid this pretreatment one? So then, yeah, this is some biological conversion to produce different products. Okay. Okay, so I was talking about what next we can do to avoid the pre-treatment. So we did some research at our institute. So I will show you some of the slides which we have done. So we mainly focus on the thermophiles. Even for the anaerobic digestion, we used or we developed a thermophilic consortium where we could avoid a pre-treatment completely. So we will go one by one. We have done a project on process development for bioethanol production from agricultural residue, where we isolated several thermotolerant yeasts and developed for further increase in bioethanol yield. Okay, so there were two isolates. One, okay, initially we named like Nire K1, Nire K3 based on our institute name. Then we characterized them and then found it is the Cluaromyces marxianus, which is thermotolerant one. So you can see it can utilize glucose, it can utilize xylose. If even in the mixture, it can utilize both glucose and xylose. But the difference is this red part is showing, I mean, it's the glucose utilization, or sorry, remaining glucose in the broth. So in that around 12 hours, uh, just a minute, my laptop battery is going down. So just give me a minute. Huh? I'm sorry, actually, I'm not in my office. I'm on leave. That's why there may be some discomfort.
It's okay,
Yeah.
sir. You continue. You are audible. No issue.
Okay. Okay. Thank you. Okay. So, just a minute. All right. Okay. So, we isolated these two thermotolerant yeast, and they could utilize both glucose and xylose, but the rate of utilization of glucose was faster than the xylose. Okay. Then we did fermentation for both glucose and xylose. Then you can see this is on glucose, so it could utilize around 10% of glucose in 16 hours. The second organism could utilize in 12 hours only. Okay, in the last slide, you can see the ethanol yield. Ethanol yield also matters in the any biological process. So it was around around 40%, but in case of second organism, it was less than 45%. And then we use xylose. So we kept 20 grams per liter, 2% xylose in the medium, and then could utilize in 36 hours. But in the second organism, it couldn't even utilize completely in 36 hours. And the rate of ethanol production and was very slow, and they also produce xylitol. So this black part is showing xylitol. In second case, xylitol production was less. Okay, then we did in the mixture of glucose and xylose. We kept 25 gram per liter each, glucose and xylose. So the glucose was utilized first, and then xylose was utilized. It was the case in both the species. Then we use paddy straw, and this is the composition of paddy straw. So there was around 40% cellulose, 26% hemicellulose, 11-12% around lignin. Silica was around 9-10%. So we followed alkaline hydrogen peroxide and sodium hydroxide. We optimized it, and then we further use sodium hydroxide for saccharification and fermentation. So we first did the separate saccharification and fermentation. So after the saccharification, whatever sugars were there, we use for the fermentation. And then you can see the ethanol yield was around 0.38 gram per gram, and then ethanol productivity 0.221 gram. Then there were some byproducts like acetic acid and glycerol. So the maximum ethanol concentration was around 4 gram per liter. Okay. Then we did simultaneous saccharification and fermentation, where we use salic CTEC2 enzyme from the Novozyme and our organism, and fermented at 45 degrees centigrade. And here you can see this ethanol red part, and the more than 25 gram per liter of ethanol we could have through SSF, which was around 4 gram per liter in separate hydrolysis SHF. So comparably, it was very high ethanol concentration we obtained through SSF. So this is the advantage. So I will disclose something. When we do separate hydrolysis and fermentation, so there is one enzyme beta glucosidase that is inhibited once glucose is accumulated. I mean, as soon as the glucose concentration increases, it inhibits that enzyme. So after some time, you will or they will be there will not be complete utilization of cellulose when we do separate hydrolysis. But in case of SSF, as soon as glucose is formed, that is utilized by the fermenting yeast, and there will be no feedback inhibition or product inhibition over there. That's why we are getting maximum ethanol concentration. Even that the fermentation time, that was very less. Almost maximum cellulose could be utilized within 8 hours, and even we ferment it up to 24 hours, but little increase in ethanol concentration was observed. So if we stop this fermentation at 8 hour only, then we can have the productivity of 3 gram per liter per hour. And if we compare the ethanol yield, so based on the raw biomass, we got around 220 grams per gram, 220 grams per kg of paddy straw. That will around more than 250 liters per ton of paddy straw ethanol. So other than that, we had some xylitol, glycerol, acetic acid as well. Now, in the last project, we observed the glucose utilization was faster than xylose, or in other terms, we can say there was a slow xylose utilization. So in the next project, we developed that yeast with the enhanced xylose utilization. So I will show you the methodology or the strategy which we used. We did the adaptation or environmental adaptation in 2 stages, stage 1 and then 2. Then we cultivated continuously in the bioreactor. Then we did the protoplast fusion with the organism, which is very efficient in the xylose utilization. And then the organism or the fused cell or fused yeast was used for the fermentation. Okay, so in this picture you can see, so we did the batch wise. The first batch inoculum was used in the second batch, and second batch inoculum was used in the third batch. So similarly, up to around 50 batches, there was a continuous increase in xylose utilization. So this is showing the percentage of xylose utilization. And then this is the dry salvate. So we observe dry salvate as the final product. And so up to 50 cycles, there was a continuous increase in both dry salvate as well as xylose utilization. But after 50 cycles, there was a stagnant increase. So we stopped it after 60 cycles, and further characterization was done for the different sugar utilization, ethanol fermentation, ethanol yield, and other parameters. Similarly, for the second organism we did, so the same pattern, almost same pattern was we obtained over here. So this first adaptation was done in the rich media, which is like we use the undefined media, which may not be economically feasible, or I mean economical at industrial scale. So mostly in the industrial scale, we use salt medium or defined medium, where the minimal salts are used. Okay, so when we use that minimal salt medium, then again we had the issue of xylose utilization. So we did secondary adaptation, where the further increase was observed when we did the adaptation, sequential adaptation. So like up to 45 or 50 cycles, there was an increase in the utilization of xylose, but further there was a stagnant, or there was no further increase in xylose utilization was observed. So we stopped there and then further characterized for biochemical parameters. So around 45 or 50% xylose only could be utilized after second year adaptation. Uh, I mean, which was not again the up to the mark which we were looking for. So we did protoplast fusion with organism which having the high catabolic activity on xylose to produce ethanol. So different methodologies were used. Okay, so the fuse cell we named like Nire FKS A1. Even I mean, during the process of isolation of those fuse cultures, we maintain the temperature like 45 degrees centigrade. But the second culture which was used in the fusion was mesophilic. Okay, so in the isolation, when we use the thermophilic or thermotolerant conditions, so the maximum characteristics were from the chloromyces only, but there was an increase in xylose utilization. So when we use that organism for the in simultaneous saccharification and fermentation of alkali pretreated paddy straw, so there was a very sharp increase in the ethanol yield. You can see here in they are the wild cultures which we isolated earlier, that showed around 144 grams per kg of paddy straw. uh, this is 1.1 showing the primary adapted, 1.2 showing the secondary adapted. So there is a little increase in the yield. Similarly for the second culture, but when we use this fused culture, there was a high increase in the ethanol yield was observed, which was around 237 gram per kg of raw paddy straw. If we see in terms of liters, it would be around 280 or something liters. Okay, another project we have done that is in collaboration with the Brazilian partner. So that was Bagasse, Biorefinery of Bagasse for ethanol and value-added products. So where we focused on ethanol along with gas, biogas, and xylitol, lignin-based products. So we follow different pretreatments like acid. We use H2SO4. In alkali, we use liquid ammonia. NaOH. So you can see here the maximum glucose recovery was observed in acid was 521 gram per kg of raw biomass, whereas in liquid ammonia it was around 545 gram per kg. So the maximum recovery of sugar was in liquid ammonia only. You can see the optimum conditions. We use FCCD to optimize these parameters like ammonia concentration, solid loading, temperature, and time. So around 15% liquid ammonia with 10% solid loading at 85 degrees centigrade for 24 hours. So this was the optimum value for liquid ammonia pretreatment. Here mainly glucose and xylose were obtained in the enzymatic hydrolysate only. So in the next slide, I will show how it performed. So this is the mass balance for acid pretreatment. So if we see 1 kg of bagasse, it contains around 40% cellulose, 24% hemicellulose, 18% lignin, and 5% ash, and 10% extractive. Ma'am, if I'm exceeding the time, then please you can indicate it.
No, you can go ahead. You are not exceeding. I can complete it.
Okay,
Continue
okay,
with
thank
that.
you. Okay, thank you. Okay, so this is the pretreatment conditions which we optimized. There were two streams, one with the liquid one and another was with the solid. So in liquid one, mostly hemicellulosic part could be solubilized while this acid pretreatment. So like in the pretreated hydrolysate, we observed glucose around 17 gram. So this is just like negligible. So little solubilization of cellulose was observed, and maximum xylose, it was around 99 or 100 grams, followed by galactose, arabinose, mannose, salobiose, acetic acid, and some other acids were observed. And in the pretreated, in the solid part or pretreated solid, there were around 378 grams cellulose. So you can see in the raw bagasse it was 400, but in the pretreated one it was around 378. So little solubilization of cellulose was there when we used this acid. And if we compare hemicellulose, so earlier it was 241 and later on it was 41 only. So around 200 gram hemicellulose could be solubilized while this acid pretreatment. And lignin again there was a little loss because you see there are two types of lignin available in the biomass. One is alkali solubilizing, another is the acid solubilizing. So the fraction of acid solubilizing lignin is very less. You can see over here. All right, and maximum loss of ash and extractives were observed. Uh, then when we followed saccharification, so around 193 grams of glucose, 20 grams of xylose, and 88 or 89 grams of cellobiose was there in the hydrolysate. And in the residual biomass, there was 93 grams of cellulose was there, 15 gram of hemicellulose, rest lignin and ash and others were there. So we can observe here the cellulose which is remained there, that is mostly crystalline cellulose, or due to the feedback inhibition of beta glucosidase, you can see this salobiose in this liquid part or in hydrolyzate. This is the maximum in the hydrolyzate due to the feedback inhibition of beta glucosidase only. That we can overcome through SSR. This is the mass balance for liquid ammonia pretreatment. So this composition is same, and if we, after pretreatment, there was a little solubilization of both cellulose and as well as hemicellulose. So this is the good part here, because I mean, if we want to have two separate streams for pentose and hexose sugars, we should go ahead with the acid pretreatment. But if we want to have both glucose and xylose in a same mixture, then we can follow liquid ammonia pretreatment. So in this part, mostly cellulose and hemicellulose were intact even after the pretreatment. And once we did the saccharification using the enzymes, then most of cell glucose and xylose were in the enzymatic hydrolysis. So this is the compositional data after pre-treatment and saccharification and even SSF. So you can see this is untreated biomass where 40% cellulose, 24% hemicellulose is there. After the pre-treatment, sorry, acid pre-treatment, cellulose increased while hemicellulose decreased, and at the same time lignin increased. These increase in cellulose and lignin content was due to decrease in hemicellulose only. Similarly, in liquid ammonia, both cellulose and hemicellulose increase and lignin decrease because most of the lignin is solubilized in alkali only. So that's why when we treated with the liquid ammonia, most of the lignin was solubilized, and there was an increase in cellulose and hemicellulose. And then when we did saccharification, then there was a decrease in cellulose, little decrease in hemicellulose, but increase in lignin. That increase in lignin was due to decrease in cellulose only. But when we did liquid ammonia pre-treated saccharification, then there was a decrease in both cellulose and hemicellulose because both were intact. So from 52 to 33 cellulose was there. But you can see here there was a little solubilization of cellulose was there. That may be due to the crystallinity of the cellulose. Okay, now we can see the difference in case of acid pretreated SSCF. There was 22% cellulose was there. At the same time, in the pretreated saccharification, 29% cellulose was remained there. So this increase in cellulose utilization was due to avoiding the feedback inhibition or product inhibition. All right. Similarly, in liquid ammonia, there was only 8% cellulose remained after the SSCF, which was around 33% remained cellulose after the saccharification only. So, using this SSCF, we could avoid the product inhibition. Okay, this is some picture showing separate hydrolysis and fermentation and simultaneous saccharification and fermentation. So we got around 208 gram per kg of raw biomass in case of SSCF. Here actually we use secondary adapted culture only. Chloromyces marxianus Nere K3.2. But when we use the fused culture, this was further increased. Okay, so this is showing optimized simultaneous saccharification and fermentation. Okay, so this was further increase. Okay, this increase in ethanol yield due to use of HTEC2. So we blended the enzyme blend of CTEC2 and HTEC2 in the ammonia pretreated biomass, and we obtained around 217 gram per kg of raw bagasse. Okay, yeah, one thing, if we go in the separate hydrolysis and fermentation, so saccharification takes around 72 hours and around 24 hours of fermentation. So almost 96 hours it takes to complete the process. But when we go with the SSF or SSCF, it takes only 16 hours. So we decreased like one sixth or 1/7 fold of the fermentation time. Okay. And we actually most of the lignin is solubilized in the alkali. So when we did alkali or liquid ammonia pretreatment, we reduced the pH up to 2, and whatever residues precipitated over there, we separated out, washed, and further treated with the acetic 2, and whatever crystals were there, they were the extracted lignin. So that was further valorized following the enzymatic process as well as the catalytic high temperature processes. And we obtain the products like vanillin, syringic acid, syringaldehyde. So these are the concentration if we see in terms of the yield. So around 64% vanillin was there, 53% syringic acid, and like 23% syringaldehyde was there. And further, the residue of SSCF we use for the biogas production. We see the biogas production, the acid pretreated bagasse, there was around 12 liters per kg of raw bagasse, and it took only 3 days. And similarly for the liquid ammonia pretreated biomass, it took 8 days and produced around 70 liters per kg of biogas. Further, the xylose available after the acid pretreatment or residual xylose available after the fermentation of ethanol. That xylose was utilized to produce xylitol, and in the feed stream it was around 25 gram per liter of xylose, and it produced around 21 gram per liter of xylitol with the 94% xylose xylitol yield. And xylitol yield actually it was 85 grams per kg of raw. This is based on the raw biomass, but it is based on the xylose utilized. And the volumetric productivity was around 0.45 gram per liter per hour with retention time of 48 hours. So you can see we have produced multiple products like ethanol, xylitol, biogas, and lignin based products. So this we can call biorefinery. If we see the mass balance of acid pretreated biomass or bagasse, so we produced biogas 25 liters, and then biofertilizer it was around 92 grams, xylitol 85 grams, ethanol 100 grams. Okay, so we had ethanol, biogas, xylitol, and biofertilizer. And in liquid ammonia, we had 217 grams of ethanol per kg bagasse, and then biogas around 18.5 liters, then biofertilizer around 123 grams. We also valorize lignin to produce vanillin, syringic acid, and syringaldehyde. We further did the energy balance. So energy balance, the total input energy was around 18.5 megajoule per kg. In return, total energy output was 5.3 or 5.4 megajoule only. So this is very less. So this, I mean, acid pretreatment may not be feasible in terms of energy. But if we talk about the liquid ammonia pretreatment, the total energy input was around 18 megajoule, which returned in terms of output of 12.87 megajoule of energy per kg, which is around 72%. So we can consider it. I mean, up to 70% if we are getting back, then the process may be energy efficient. So, based on above or earlier slides, we can conclude like cellulose, hemicellulose, and lignin. We can have products like ethanol, xylitol, biogas, vanillin, syringic acid, syringaldehyde, biofertilizer. Okay, we also done some biophysical characterization. So this part was done in Brazil only. So we did XRD for the crystallinity check. So in raw bagasse, there is some crystallinity like 49%. With acid pretreatment, it increased, while with the alkali pretreatment, it decreases. Once the acid pretreated biomass was saccharified, so mostly amorphous cellulose is solubilized while saccharification. So crystallinity increases. Similarly with the alkali pretreatment. And then if we see in after the SSCF, the alkalinity will be very high. So this is the confocal laser scanning microscopy. This is basically used to check the lignin component. So the first picture showing lignin structure in the raw bagasse. So after the acid pretreatment, you can check change in the color. That this color is due to the like maybe redistribution or change in the structure of the lignin, so it gives the different color. Similarly, in the C part, this is enzymatic hydrolysis. I mean, after the enzymatic hydrolysis, there is a little change in the color as well. And this D showing in the ammonia pretreatment. So in ammonia pretreatment, actually most of the lignin is solubilized. So you can see the like destruction of that lignin. You can see in this picture. So similarly, in like is enzymatically saccharified and SSCF, you can check. Okay. Okay, this is the field emission scanning electron microscope images. So this is for the raw bagasse. So you can see the smooth structure of bagasse, but once it is pre-treated, there is a rough surface and change in the surface you can observe over here. So this is B showing the pre-treated with acid, and then this is with the ammonia. So there is a almost complete destruction of the surface of biomass. You can see. Similarly, other pictures also you can see here. This is the NMR spectra. So you can see this black digit mark, they are showing cellulose, blue, red one showing the hemicellulose, then blue one for the lignin. So after the alkaline or alkaline pretreatment followed by saccharification, you can see this there is a no change in cellulose and hemicellulose, and even in the this hemicellulose. But once it is saccharified, the cellulosic peaks also reduced due to its solubilization. Similarly, with the acid pretreatment, you can check this is the raw SCV, pretreated SCV, and once it is pretreated, mostly that hemicellulosic part is solubilized, but there is a little decrease in the cellulose was observed. Okay, now we further developed a consortium for biogas production, which we discussed in the last slides. where we propose to have a solution for to avoid pretreatment. So we focus on the development of thermophilic consortium. So we targeted temperature between like 50 to 55 degrees centigrade, and we developed the consortium which is grown on the water hyacinth, and we use around 10% solid loading at 50 degrees centigrade, and it takes 30 days, around 30 days to get it developed. Even in the water hyacinth, it produce this much of around 370 liters per kg of biogas with 60% methane. When we tested this water hyacinth, I mean consortium grown on water hyacinth, where we use 10% seed, 5% solid loading, and we optimize actually this temperature, which was around 52 degrees centigrade, and it took only 12 days to get it digest most of the paddy straw. If we compare this with the conventional one, I mean, in conventional, we basically use cow dung. So if we digest paddy straw with the cow dung, it takes around 40 to 60 days, even more. And the biogas yield again is very low in case of cow dung, but here we got around 330 liters per kg with the methane content of 57 or 60%. Okay, this is a flow diagram showing treating 30 kg of paddy straw per day, and out of that 30 kg, we can produce around 15 meter cube per day of biogas with the 60% methane content, which having the capacity to generate electricity of 21 kilowatt hour per day, or we can have cooking gas or bio CNG of 4.3 kg per day. The similar consortium was tested on the grass. So in grass also we use 5% solid loading, 10% seed at 52 degrees centigrade, and it took only 16 days. And biogas yield which we obtained was 700 liters per kg with 60% methane content. And this is the picture for treating 400 kg per day cow dung, sorry, grass, and it having the potential to produce 174 meter cube per day with methane content of 60%. Out of this biogas, we can generate around 350 kilowatt hour per day of electricity with cooking gas or bio CNG 70 kg per day. And this is showing biogas production on vegetable waste. Just okay. In vegetable waste, we used actually there was around 88% solid were there. So we sorry 88% moisture was there. Around 12% solids were there. But by adding some water, we maintain 8% solid loading because this we actually optimized, and seed percentage also we optimized, and temperature was 52, and it took around 25 days to digest it. But the beauty was in the first phase, around 5 to 7 days, it produced hydrogen only. Okay, once hydrogen is over, and there was a accumulation of acids in the liquid, and once pH was maintained, it started producing methane. And throughout the, I mean, after that, they produce only methane rich biogas only. So if we see the biogas yield, so it was around 250 liters per kg of hydrogen rich biogas, while 550 liters per kg of methane-rich biogas. And hydrogen content was around 35%, while methane was 65%. So if we have such, mixture of hydrogen and methane, that we generally call it bioethane. So basically, if hydrogen is there, so you will have the high calorific value. Or the government of India also accepted to use hydrogen-rich CNG, or we call HCNG in the transportation, because it having the higher calorific value than the pure methane. Okay, so I will conclude over here. There is a great potential in lignocellulosic based biorefinery. The products like bioethanol, biogas, they may be the major products, and which having the capability or potential to replace fossil fuels, and having applications in transportation, heating, power generation, like that. Other products like xylitol, furfural, hydroxymethyl furfural, lignin-based products, biofertilizers, even secondary products we can generate, and it having the potential as a byproduct, and we can utilize in a circular bioeconomy, and we can develop a zero waste discharge technology out of it. Okay, and agricultural waste can be, best managed to avoid uncontrolled degradation or burning using this biorefinery. So let's join hands together for the sustainable development of society, for energy, environment, and economy. I acknowledge Ministry of New and Renewable Energy, Department of Biotechnology, SCRB, and my own institute, Sardar Swaran Singh National Institute of Bioenergy. Thank you.
Thank you very much, sir, for your presentation and detailed knowledge of biodiesel and the green initiative which your institute and ministry is taking. Certainly, this will encourage the people to go towards the biofuels and encourage people in research project. Now, I request the participant and dignitaries, if you have any conversation or queries with sir, you can go ahead, as it is not possible to have such a eminent personality every time on a FDP platform for an invited lecture. So I request the participants and the entities, if you have any queries or conversation, you can go on with your talk.
Hello.
Yes, please. Yes, ma'am.
I'm asking for the participants to ask questions. If anyone like to ask question. No questions from the participants. May I ask you questions?
Yeah, please, ma'am.
Sir, there is a lot of policy changing taking place at the government level.
Yeah.
So, but still bioenergy and biofuel production in our country, despite the fact that we have so much of biomass material available throughout the year, why it is not picking up? What is the problem? That we are facing? Since you are working at such a place which is National Institute of Bioenergy, but still, we are even at the government level, or even at the policy level, we are focusing more on solar energy and wind energy. But at present, there is not much focus on this biofuel. And since I come from the same field, so I would like to see emerging focus on this particular form of renewable energy. So can you briefly tell me
Sure,
the
ma'am.
future aspects?
Thank
of
you for the
this?
question. Yeah, it's a nice question. So, we need like stakeholders, or the businessman to come into this field to take up the research or the process or the technology which we have developed.
Mm-hmm.
So any businessman, I mean, first question come, how much investment would be there, right? So
Yes.
yeah, that is the first question came. Then later on he thinks about his profit or something. What is technology? Technology comes secondary actually. So the investment part, if we talk about this ethanol one, it's like
Mm-hmm.
there are multiple steps involved. So multiple processes are involved over there, like pre-treatment, then saccharification, fermentation, then recovery, then secondary recovery, I mean distillation, then secondary distillation. So there are multiple steps, and you need different reactors or the equipments to process at each step. So it takes like huge investment over there. Uh, Once I completed that project, I thought we should go for the pilot plant. And I asked for some quotation from the premier agency and I mean, industry. It was such a huge amount. And I mean, if I submit for the funding, they will
Uh,
also refuse it because government also, I mean, they can't invest in for the pilot plant, but that can be done if we go with the like public private partnership or something. But the business should also be there, and even it should be the techno economically viable. So it is
yes.
like
That is I think economical viability is the first and
yeah,
foremost thing that is, I mean, becoming a hurdle in this particular form of renewable energy.
right. So so techno-economics can be like if we go for a single product that cannot be technically or economically viable. So that biorefinery concept came into the picture. People thought to produce multiple products. So
Yes.
wherever there is a requirement of ethanol, then they can produce ethanol. If I mean there is no requirement or the demand is low for ethanol, then they can produce some organic acids. They can produce
Okay.
even biogas. The investment in
Mm-hmm.
biogas is very less than the this ethanol one. But I
And
mean that investment can be overcome or can be combined in a collective manner, like ethanol production, biogas production, xylitol production. Xylitol is very demanding in the market. Even
yes, yes.
yes. So and
Uh,
the lignin basically either we use in the combustion or we just throw it. That can again be
mhm.
valorized to multiple products. The products which I discussed in the presentation, like vanillin, syringaldehyde,
Yes,
guancol,
yes.
they are actually
Uh,
very demanding in the market. So
mm-hmm.
and government also promoting for the zero waste technology. So in such a
Yes.
manner, we can develop that biorefinery. And again, the incentives or the support from government is always needed. Uh, there was a
I think that
yojana,
is also
Pradhan
lacking
Mantri,
somewhere.
yeah, Pradhan Mantri Jan Yojana, they also announced, it was also announced to support like some 5 or 6 biorefineries to set up in the
Mhm.
country. But I don't know what is the status, but it will come up. Even 2 biorefineries are coming up in the Panipat. But
Okay.
you know, this is just actually there are very few people, or actually there is no role model or role industries available in the country, even in the world. So
Mm.
the businessman gets afraid to come into this field. Once one success story is there, then I think it will come up or it will flourish. Similar case was with the solar. Earlier, the cost of electricity production by solar, it was very high. But once
Mm.
like people started using, people believing in it, Now the cost is even lesser than the conventional one. So
Yes.
yeah, people's interest required government support. I mean, industries interest, public support. They all need it to get it flourished.
Yeah, because since you are in the, prime position in a government institution to push this bioenergy sector. Recently, Prime Minister has also announced about the co-generation of this bioethanol with the biogas in the sugar
Yes.
factories or
Yeah.
distilleries. So I think that could be a plus point for increasing the production of ethanol, because
Yeah.
for this not much infrastructure is required, I guess.
Yeah. Right.
So you answered my question very well. Thank you.
Thank you. Thank you, ma'am.
Hello,
Any
ma'am.
question from? Yes, go ahead with your question if you
Yes.
have.
Yes, sir. My name is Jishnu, sir. Sir, I have one question.
Yeah.
Like, sir, like from my BTech time onwards, I've seen that there are a lot of research going in biodiesel development.
Okay.
And as you said, practically, when we operate in the available engines, there are a lot of issues like clogging, maybe the cold starting issue like that. Sir, as a mechanical engineer, I feel that along with the biodiesel development, actually we should develop engines that run smoothly on biodiesel, right, sir? There is also a requirement
Yeah.
of
Yeah. Definitely. Yes. Yes. Yeah. Right.
I think there is a need
So
to
is
modify
this your
the
okay? Okay.
engines.
Okay. Let me finish. I thought you were asking another question. Okay. You know, up to 10 or 20% blend, there is no need to modify any engine. Okay. And one more thing, there are specifications available for biodiesel which need to be followed. If you do not follow, there may be issue with the engine, right? Another thing, if you are going beyond the limit which is decided through research, then of course you will have issue with the engine. So you need to modify engine if you want to go, if you want to blend the biodiesel with the beyond that limit. Is it all right, Jishnu?
yes, sir. Sir, sir, if you look at the automobile engines, these petrol and diesel engines, actually they have been optimized through some 100 years of research or more for suiting the present petrol and chemical technologies. So if we look at, like, sir, we are doing a research on biodiesel, but the problem is we are always trying to make it suit with the available engines.
Okay.
So when we have an opportunity to produce an engine itself, which will operate better on the higher percentage of biodiesel, where we have a higher percentage of biomixed component over the diesel. So then actually we are given a autonomy to work freely on this research rather
Yeah,
than
yeah. I guess you are a mechanical engineer. So if you have any engine with you or with your lab or institute, then I guess you can ask for the liberty to change anything needed to modify that engine. Actually, I'm not a man of engine, so I can't comment over there because I'm a biochemical engineer. So I guess, yeah, you can work on in its modification in terms of exhaustion, in terms of energy efficiency, in terms of blend, how much you can increase or decrease, so that you can optimize. And you know better
I
how.
have the same.
Yes, ma'am.
You continue.
Yeah, I just finished. No issue.
yes,
Okay.
ma'am, my doubt is cleared, ma'am. Thank you, ma'am.
Okay, I think I have the same opinion that we have to do some modification, or the other part related to this is where we can do a lot of research and development is the processing of biodiesel. Basically, refining and processing we are lacking in our country. And if you go to European countries and other like US and all, they have a advanced technology related to refining and processing of biodiesel as well as bioethanol, and that is why they are promoting these type of biofuels a lot in their countries. But we should also do this because we have a lot of biomass, we have a lot of waste material, agricultural waste particularly, that is causing unnecessarily the pollution load in our atmosphere. We can all reduce by simple use of this technology related to refining and processing of these biofuels. So, Sachin can help us in doing this.
We collectively can do something.
Yes, we can do this. And we hope that one day we will get the success of getting 100% biodiesel or 100%.
Success stories in front of us, like solar. Earlier, nobody believes in solar, but nowadays solar is booming, right? So similarly, we have to brought up this, biomass.
Yes. Okay. Any more questions from the participants?
Dr. Chelia, he's raising hand. I guess hearing
Yes. Go ahead, Dr. Chelia. You can ask your question. Hello. Is there some problem related to audio?
No, ma'am. There is no problem related to audio. Nobody else is even typing question in the chat box also.
Okay. Oh, he has dropped his raising hand. So I guess he has no question. Yeah.
No. So yes.
Any more questions from the participants before we ask our speaker to leave the meeting? I think no more questions. Thank you, Sachin, for a wonderful and elaborative talk. It was very insightful for all the researchers who are interested in bioenergy. You are doing a great job. Very elaborative research you are doing. Thank you. Thank you very much.
Thank you, ma'am, for inviting me. It was great pleasure to be here. Thank you. Bye.
Thank you. You can leave the meeting now.
Thank you, Dr. Sachin Kumar and Dr. Neelam Mishra for their insightful views on bioenergy and biodiesel and the research upcoming in this field. So hope that one day it's future to see that bioenergy really dominates in the energy industry, just as solar is booming right now. Now I request the dignitaries and the participants that if you have any queries regarding the feedback form or examination, because the examination form and the assessment form is going to be sent to you via email also, and the link will be shared on the group, WhatsApp group also. So you will be given a duration of the lunch up to fill the form and mail it back to us. So kindly do the needful, and we will join you in the next session, 2:30, by Dr. Bhupen Singh, who will speak on the mental health of cognitive wellness, because mental health is the ultimate aspect of mind which promotes us to do all the ideas and go forward in life. Because if mental aspect is not good, we cannot do anything big, or we can not achieve our goals. So kindly be patient with us, and thank you for your cooperation and support till now. And we will be back at 2:30. Yes, ma'am.
Hello, sir. Yes, sir. Can we have the test now?
Yes, you will have the test now. We will be mailing it to you on the email, and I am sharing the link on the WhatsApp group also, so you will be able to.
Yes, sir. Thank you.
Thank you. And we will meet for the next session on 2:30 p.m.
Okay. Sir, what is the duration of the test?
Duration you have to submit it by lunch. Now it is 1:43. You can submit up to 2:30 p.m. Or
Okay,
even then,
okay.
if you are require, you can 10 15 minutes extra. That is not
Okay, thank
much of
you.
it. Thank you. So we will meet at 2:30 p.m. and we will send the assessment, feedback, and examination link on the WhatsApp group and mail it. And we will do the next. So I am closing this session now, and we'll meet at 2:30 a.m. with Dr. Bhupen Singh for his lecture on mental health of cognitive wellness. Thank you very much for your patience and assistance, and hope to meet you at 2:30 p.m. Thank you very much.